Hello, everyone, and welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Josh York, and I'm a postdoc at Northwestern University, and I'll be moderating today with Diana Fulmer, a postdoc at UPenn. We are excited to highlight the work of our outstanding postdoctoral members here at SDB. Today, Talia Hatkevich from Duke and Gerrald Lodewijk from UCSC will share their research. Each speaker will give a 20-minute talk, followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. It's my pleasure to introduce our first speaker today, Dr. Talia Hatkevich. Dr. Hatkevich earned her PhD in genetics from UNC Chapel Hill and Jeff Sikelski's lab, where she studied mechanisms regulating various events during meiosis, including chromosome pairing and meiotic recombination, using Drosophila as a model system. She's been a highly productive scientist in her field, publishing in top-tier journals such as PNAS, Current Biology, and PLOS Genetics. She is currently an NIH-funded postdoc in Blanche Capel's lab at Duke, where she uses mice to investigate the development of male germ cells. Talia, thanks for being with us today, and I'll hand it over to you.
Thank you so much, Josh. Let me share my screen here. All right, and thank you everyone for tuning in today. And as Josh mentioned, my name is Talia Hatkevich, and I am a postdoc in Blanche Capel's lab at Duke University. So I just want to start off by talking about one of the most fundamental events in biology, and that is the passing of information from one generation to the next indefinitely. And this allows for evolution, the continuation of species, and really even just passing on the family name. This information is passed on through the germ cell, which is the sex cell of the animal that will eventually give rise to the mature gamete, to the sperm or the egg. And the information that's passed on indefinitely is both genetically and epigenetically packaged inside this germ cell. So an inherent question that the immortal germline poses is, how is the integrity of information maintained? And, this question is particularly relevant within the context of the mammalian male germline. And I will tell you exactly why. So the male germ cells must undertake a momentous journey to establish the male germline to eventually pass on their genome. And in male mice, which is the model I use, this starts at embryonic day 6.5 or E6.5. This is when the primordial germ cell is specified. Then the germ cell has to actually migrate through the embryo to enter the native gonad. And then it gets settled there at E10. And then at E11.5, the germ cell is specified into a male germ cell, or MGC. I want to point out that this is also referred to as prospermatogonia. However, I'm going to refer to male germ cell as MGC throughout this talk. After days of proliferation, the MGCs will then enter or exit the cell cycle at embryonic day 14.5. And they remain in this G0, G1 mitotic arrest for about 7 days, where at P1, they re-enter the cell cycle. So postnatal day 1. And they re-enter the cell cycle, essentially emerging as spermogonial stem cells, or SSCs. And I like to think of SSCs as the biological cell that has won the most coveted prize. And this is because SSCs are this resident stem cell population within the testis that can both differentiate and self-renew. So for the entirety of a male's reproductive lifespan, it is able to provide the progenitors of sperm, thereby passing on whatever is in their genome to the next generation. Then after the establishment of SSCs, we get to mature sperm. So not only is the male germline going through all of these developmental steps from when it's specified at E6.5 to when the SSCs are established at P2, but they're also going through unique molecular events that happen only in germ cells. And some of these events that occur during this time is complete demethylation of the genome. So this happens at about 9.5, and the genome is hypomethylated by about E13. And then de novo DNA remethylation occurs, starting at about 15.5 by birth. We get hypermethylation finally. So also facilitating with that DNA methylation and remethylation, we also have chromatin rearrangements that are unique to germ cells. And we also know that germ cells undergo hypertranscription. And so this means that germ cells exhibit transcriptional levels that are up to 20 times higher than the somatic counterpart. Right, so there's a lot going on developmentally and molecularly that are unique to the male germ cell. And so to maintain the epigenetic and genetic integrity, the germ cell must employ some regulatory mechanisms. However, many of these mechanisms during development are unknown. So today what I'm going to do is I'm going to talk to you about two things. First, I'm going to tell you a story I've been working on during my postdoc for the past couple years that I hope to be published soon. And then also a program that, a branch of my program that I am currently developing. So let's get to it. So this G0, G1 arrest of the male germ cells is actually vital for development into the SSC fate. So what's happening is that the pluripotent germ cell identity is actually being faded away, and then SSC stem cell identity is really being acquired during this time. And what is actually having this acquired is de novo DNA methylation, as I talked about. So without the de novo DNA methylation occurring properly, the establishment of SSCs does not occur. Right, and then also, so helping and facilitating with the DNA methylation is the histone landscape. So this histone landscape is rapidly changing in response to the remethylation of the DNA. So my overall question for this particular project is, how is this rapidly changing epigenetic landscape regulated, ensuring the development of the MGCs into the SSCs? And I'm going to answer this looking through the lens of an RNA binding protein called DND1. So DND1 is a vertebrate-specific, germ cell-specific RNA binding protein that is important and vital for the germline stem cell or the germline development. And here we know that DND1 is highly expressed within the G0, G1 MGCs. So we created a mouse line that endogenously tags DND1 with the GFP. And using this, we can actually see here that germ cells at E16.5, which are all of these cells here, are expressing DND1, and it's all over the cell. So it's within the cytoplasm, the nucleus, and within these puncta, which are germ granules. We also know that DND1 binds hundreds of transcripts throughout the G0 period, and many of these transcripts encode for epigenetic regulators. And we know this because a previous postdoc in the lab performed a RIP-seq study using our DND1 GFP line. And one of the epigenetic regulators that binds, whose transcript binds DND1 is SETDB1, which is a histone methyltransferase. So here is our RIP-seq, and you can see here that compared to input, there is no enrichment of the RIP-seq at E14.5 of SETDB1 transcript, but there is at E16.5 and E18.5. So I'm going to answer my question by posing the hypothesis that DND1 regulates the protein expression of its target transcripts that encode for epigenetic regulators, and this happens post-transcriptionally. And I'm going to use SETDB1 as an example today. So the first question that I want to pose is SETDB1 actually subject to post-transcriptional regulation? And this is actually a really hard question to answer in our system because we can't do a lot of molecular analysis because we don't have any cell culture or anything. But what we can do is we can look at the correlation between transcript levels and protein levels to then infer something about post-transcriptional regulation. So by looking at SETDB1 protein throughout G0, i.e. 14.5, 16.5, and 18.5, we actually see that the protein decreases significantly throughout the 3 stages. So it decreases about 60% from 14 to 16, and then about 50% from 16 to 18. This is quantified using FACS analysis and looking at the fluorescent geometric mean of SETDB1 protein. Right, so this is also seen here when looking at the SETDB1 localization within germ cells. So here I just want you to look at the you can just look at the SETDB1 panel. So at E14.5, we see that SETDB1 is localized specifically in the nucleus. And then at 16.5, we actually see this really interesting pattern where SETDB1 starts to actually be expressed within the cytoplasm. And then at E18.5, SETDB1 is seen in a punctate manner, both within the cytoplasm and within the nucleus. All right, so what about the transcript levels? Well, using qPCR, we see that there is no difference between transcript levels between 14.5 and 16.5. This is interesting simply because at 16.5, we see a strong reduction of protein levels. So this in itself is suggesting post-transcriptional regulation of the SETDB1 transcript. Importantly, at 16.5, this is when DND1 is binding SETDB1. We see a strong reduction of SETDB1 transcripts, i.e. 18.5. However, post-transcriptional regulation of SETDB1 to regulate protein expression would suggest perhaps that maybe even though the protein is being reduced during G0, that the transcript is actually still being transcribed. So we actually just looked at this by looking at transcript localization of SETDB1 in germ cells throughout G0. And we see that we did this using single molecule fish, and we see that SETDB1 is localized to the nucleus at all stages. Because mRNAs are not sequestered or stored in the nucleus, this indicates that SETDB1 is being actively transcribed at all 3 stages. And when you look at the percentage of SETDB1 localization per cell, it's it doesn't change the localization within the nucleus at all. So this suggests to us that SETDB1 is actually being transcribed all throughout G0, even though the protein is being decreased. So this suggests to us that yes, SETDB1 is in fact being regulated post-transcriptionally, but is it actually being regulated through DND1? So to answer this question, we collaborated with Atusi Suzuki, who has a DND1 flox mouse. And so we created this DND1 mutant mouse by mating these two together with an OCT4 Cree-ER. We tamoxifen at 13.5. And then at 16.5, I was sent the samples and I analyzed SETDB1 proteins via IF at 16.5. And this is from a paper that Atusi previously published showing that if you tamoxifen at 13.5, you get a strong reduction of DND1 at 16.5. So I just want to say, when looking at the mean fluorescence of SETDB1 via IF quantitation, we actually see a significant decrease of SETDB1 protein in this mutant. This was actually really surprising to me because my hypothesis was the complete opposite, that DND1 is promoting the degradation of the transcript. But because of this, it actually suggests that SETDB1 is playing a protective role of the transcript. So let me just show you how I'm actually interpreting this. So SETDB1 at E14.5, it's not bound at all, and then we get high translation of the transcripts. 16.5, how I see this is that maybe perhaps some of the transcripts are being degraded, but DND1 is binding to some, preventing that degradation, and that's resulting in some protein. And we also see this happening at E18.5. So the mutant, you would get rid of DND1, and therefore you would have a reduction of the SETDB1 protein. Right, okay, lovely. But if SETDB1 is reduced too early in MGC development, then what is the consequence? this I'm actually asking, is this protective role of DND1 really important? So to answer this question, what I did is we created a knockout mouse of SETDB1 specifically in the germ cell using a Cree-ER system and germ cells specifically with the germ cell specific driver. Similarly to the DND1 knockout mouse, I tamoxifen at 13.5 and analyzed at E16.5. Now, this led to a mosaic knockdown, which is pretty common, but through analysis, we see that about 50% of the germ cells are affected. So just gross morphology analysis here at E16.5, this is our wild type control. We can see that nice testis cords and germ cells are marked with this TRA98. So it's a nice round testis with nicely organized cords. Now, the SETDB1 mutant morphologically at the tissue level does not look very different. However, let's look a little bit closer. So here we're looking at wild type. We're looking at testis cords marked by TRA98 which are marking the germ cells. And you can see that the nucleus of the germ cells are beautifully round and it's very consistent. Our SETDB1 mutant looks completely different. The TRA98, which is a germ cell marker, is not marking nearly as well, indicating some unhealthy cells there. And you see that the nuclei of the SETDB1 mutants are actually very abnormal and not round at all. To try and quantify this, what I did is I took the nucleus of all the diameters and I took the largest nucleus of each cell and then compared between wild type and SETDB1 mutants. And we see a significant difference. And we see a new population that's really large, but then we also see some germ cells that are pretty small, and you can see here. So I also wanted to ask, well, if these cells are unhealthy, are they undergoing apoptosis? And a marker for apoptosis is gamma H2AX, which marks double-strand breaks, but it would have to be completely nuclear wide gamma H2AX positive for it to be a dying cell. So we quantified the percent nuclear wide gamma H2AX germ cells per cord analyzed, and we see a significant increase in SETDB1 mutants. So this is indicating to us that these cells are most likely dying. But a question is so this is at the MGC level that we see this defect happening, but can SSEs actually be established? So to ask this question, we looked at testes at P4 in wild type and SETDB1. And you can see here, this is the cross section of a testis. And we're again using TRA98 to mark our germ cells. And our SETDB1 mutant, we see no germ cells in the left testis. This indicates that SSEs are not established. But what's really weird is that in the right testis, we get plenty of germ cells. This actually looks normal morphology wise. And even at 6 weeks, this phenotype persists. So at 6-week-old pups in our wild type, we have left-right testis. In our mutants, we really see the defect only in the left testis. We have no idea for this. I would love to talk about it in Q&As, and if anyone has any ideas, I would love to hear it too. So overall, with this story, so what we see is that SETDB1 is most likely being actively translated at 14.5. At 16.5, it's time for the protein to be reduced, but not too much and not too quickly. So we see DND1 coming in, binding SETDB1, and ensuring that there's still some protein that's being made. Thereby, throughout 14 to 18, we're seeing just a gradual reduction of the protein. And this in its own right is setting up a proper genetic landscape to ensure that MGCs develop into SSCs. So the next thing I want to talk about is what I've been working on developing for another arm of my research program, and that is stability in MGCs during this G0, G1 developmental time. So DNA is constantly exposed to sources, both endogenously and exogenously, that damage your DNA. It creates, the average is about 200,000 lesions per day. And the exogenous sources are UV, IR, and then also just chemicals that you come in contact with. Endogenous damage includes reactive oxygen species, things simple as transcription, replication, and even removing and adding methylation to DNA
1 minute.
will create double-strand breaks or damage. And double-strand breaks, or DSBs, are the most deleterious form of the DNA lesions. And so there are double-strand break mechanisms that are able to be to come in and repair those double-strand breaks. And the two canonical pathways are homologous recombination, or HR, or non-homologous end joining, or NHEJ. So NHEJ is thought to be like the quick and dirty repair mechanism for double-strand breaks. And this is because a complex will come and just kind of clamp on the ends of the double-strand break and then just ligate them together, so smush them in. And here we get genetic information lost. However, this is able to repair quickly, so there's a give and take with using NHEJ. Whereas with homologous recombination, it's a lot more complicated and involves a lot more proteins. However, no genetic information is lost because you are synthesizing off of a homologous template to get the information back that's lost during the double-strand break. Now, the pathway in mammals that is used, NHEJ versus HR, is largely dictated by the cell cycle. So in the cell cycle, in G1, right, you are not replicated. Your chromosome is not replicated. You do not have a sister chromatid. However, in S phase, you are getting synthesis. In G2, you have a sister chromatid, right? So this means that in G1, you don't have a homologous template readily available. So the cell will usually use NHEJ to repair. And then the cell will use the homologous recombination when they have their sister chromosome or sister chromatid nearby. Right, so this sets up a paradox in the germline. Using NHEJ to actually repair the germ cell in the genome will lead to genetic deletions, which can be then codified into the heritable germline. And this is really interesting because during the arrest, there is a G0, meaning that there is no sister chromatid nearby. And during this time, there's hypertranscription, DNA methylation, and nuclear arrangement, all which accost the DNA and causing double-strand breaks. So the question is what is the mechanism by which the G0, G1 MGCs are actually repairing double-strand breaks? So this is something I'm developing, and I have preliminary data. I just don't have time to talk to you about it, but we can talk about it later in Q&A. So going back, how is the integrity of the information within the immortal germline actually maintained? Well, genetically, my preliminary data suggests that it does it by using unique DNA repair mechanisms. And epigenetically, it is through highly regulated expression of epigenetic factors. And this is facilitated by proteins such as RNA binding proteins and DND1. And with that, there's a lot of people I would like to acknowledge. Blanche, for being a lovely mentor. I appreciate everything that she's done for me. Kayla is an undergrad from University of Richmond who has quantified a lot of the microscopy data. And then also Atushi, who is gracious enough to share the DND1 conditional knockout mouse. And with that, I'm happy to take some questions.
All right, thank you, Talia. That was a fantastic talk. So just a reminder for everyone, if you have questions, you can input them in the Zoom Q&A box, and I will give those to Talia. And then any unanswered questions we can answer later in the Q&A box. I'd like to begin actually. So Talia, I had two general questions. So one, have you considered genes, the asymmetric testis phenotype,
Mhm.
genes that play a role in left-right organogenesis? So I was just curious, like, have you considered like PIDX or lefty or nodal? Those are genes that like place heart in different sides of the body, and are you thinking those genes are involved in this asymmetry?
Um, I honestly, I don't know. We don't know the reason for the asymmetry, but lefty is actually an interesting gene to think about because it's highly expressed in the developing germline. So, I should perhaps check that out. Um, I will say that one one thing that is different between left and right is that there's been a paper is in that there's more teratomas in the left testis than in the right testis. And Blanche actually showed a couple years ago that this correlates with differences in vascular architecture. So my hypothesis right now is that perhaps the left testis cannot recover as greatly as the right testis simply because there's too low of oxygen or energy.
Mhm.
Um, but who knows? But thanks for the question. Yeah.
All right, one more follow-up, just a general question. So for PGCs and GCs, is there evidence that the gene regulatory networks involved in those, how similar are those to other types of pluripotent cells in the embryo? Are they very different? Are they sort of similar in many ways, evolutionarily conserved? What's your take on that?
Um, I I honestly don't know. I will say that the germ cell is very unique in that it can turn in at that stage, going from PGCs to MGCs, it's highly pluripotent. So it has to shut down a lot of other pathways or all the pathways to be able to develop into the germ cell. So I would suspect that it's probably different. There's a lot of germ cell specific genes that are expressed during this stage. Um, so I would say it's probably specific to the germ cell.
That's all for me. We do have a question here from Teresa Gross-Thieving. She asks, what exactly is the function of SETDB1 and how is its function affected by its dosage? That is reduced but not eliminated by DND1.
Um, Great question. So SETDB1, what it does, so it's a histomethyltransferase and it deposits H3K9. Well, it deposits trimethyl onto H3K9. So what that does is that is supposed to reduce the expression of genes and create heterochromatin. So it's a heterochromatin promoting mark. How SETDB1 is actually being affected, um, how this mark is being affected by SETDB1 during this stage in germ cell development is unknown. One of the things I would like to do is to do a ChIP-seq for the mark that SETDB1 lays down to see the developmental progression of that. But that field is rather crowded, and I think someone's probably going to get to it before I do. Um, Now, why would it need to be reduced? And how would that change the function of it? I'm guessing perhaps maybe to maintain that mark after or while the DNA is being remethylated. But again, this is all speculation.
We have another question here from Michael Layden, who asks, do you see the same phenotypes for RNA and protein levels for genes other than SETDB1? Is
Yeah.
DND1 always protective?
So we do see a similar, well, okay, so the transcripts that DND1 binds to, it can be protective, or it can actually promote the degradation of. So SETDB1 is known to have both of these roles. The genes that we see, or the transcripts that we see, a lot of them do not have the same patterning that SETDB1 does have. Some will increase, indicating that DND1 is promoting translation of, and these are more analyses that I have done that will hopefully be in the manuscript that I publish.
Looks like we have time for one more question. We have from Hector Perez, who says, "Hi, awesome work. Did you find aberrant expression of gonadal somatic cell markers in the left testis? Is the lack of GCs modifying the developmental morphology of the testis, or is this modification only attributable to mutation?"
I think that the lack of GCs is what's really leading to the morphological defect of the testis. And in the left testis that has no GCs, in the SETDB1 mutant, we have not done any RNA sequencing or anything with that. That again is a future direction that I would love to study. But that can provide some insight onto perhaps what is really going on the transcriptional level.
Excellent. Okay, I think that wraps up time on our end. Again, thank you, Talia. Fantastic talk. And I'll hand it over for the second moderator.
Thank you, Josh. And hi, everyone. I am Diana Fulmer from the University of Pennsylvania, and I now have the pleasure of introducing Gerrald Lodewijk, a postdoctoral researcher from the University of California, Santa Cruz. Gerrald joined the Ali Sharidi Lab in 2020, so he is a pandemic postdoc, much like myself and many of us. And he joined his current lab after earning his PhD from the University of Amsterdam under the supervision of Frank Jacobs. Gerrald has won numerous awards, including a recent GAN mentor fellowship from GeneTech. And today he's going to share his latest work on the self-organization of embryonic stem cells into reproducible embryo models. So take it away, Gerrald.
All right, thank you for the introduction. And yeah, I've been a postdoc here for a few years, started in the pandemic, as you mentioned, and yeah, still going. So I want to talk about a project that recently got published and highlight a couple aspects of that in the time that has been given by the organizers. So thank you for inviting me and giving the opportunity to talk here. So how about engineering preferred cells to generate embryo models in vitro? And just to segue a little bit of the previous talk, so life starts from a humble beginning, which is one cell. And this is the sperm that's fertilized the egg to create a zygote. This is the one cell, which in mammals, placental mammals, will divide to generate a more advanced stage of the embryo. So coming from this one cell, dividing to make 2 cell, 4 cell, 8 cells, so on and so forth. So eventually, you create a structure called a blastocyst, which is highlighted by the formation of an embryonic cavity and differentiation into a few different cell types. So first there is trophoblast cells, which generate the placenta, which helps to implant into the maternal uterine wall. There's the hypoblast or primitive endoderm, which generate the yolk sac and the amnion, so another supporting tissue. And the inner cell mass or epiblast actually forms the tissues of the adult body later on. So sort of all the tissue as we are, sitting and listening here. And so this pathways are generally like quite the same between species, but you can also see that there are differences in how these cells are organized. In human, you see this more like disc-shaped structures of the cell types, whereas in the mouse, you get this sort of like cylinder structure. Even though the cell types are similar, they organize in different ways. And for this talk, I'm mostly focusing on this mouse development, and especially this sort of like early implantation stage between sort of day 5 and day 6 as part of this initial study. And I'm very interested in this to find out, like, well, how does this one cell know how to form a whole embryo? So you have this one cell that forms different cell types. These cells then collectively like migrate or divide. So how is this from a fundamental aspect? How does this sort of collective and individual behavior reproducibly lead to this development of an embryo of like mouse or human or other species? And also from, I think, a more of a clinical perspective, where we know that in human, about 30% of every sort of early embryo fails to implant, leading to early pregnancy termination. And we don't really know why. There's not really a lot of information on what's going on. But it seems that trophoblast cells would have some kind of role in this because they are the ones that facilitate the implantation. So that led to my interest in like developing some of these tools and models to make sort of embryo-like structures in vitro, sort of model some of these aspects from both a fundamental and clinical angle at some. When you compare development across species, I think in developmental biology, there's not just work done in mouse and human, but also in frogs and fish and other species, and especially species that do develop in the egg, like in this case, the zebrafish in the middle here. The advantage is you can take the egg and sort of image the whole process of development relatively easily, whereas you deal with a mouse or a human, there's like all kind of ethical restrictions, but also the embryo is actually deep inside of the maternal organism, so you can't access it very easily. So I think that's another reason to use some of these embryo models to sort of scale up some of the developmental studies we're doing in both a mouse and human context. And so yeah, just coming back to some actual like work. So basically in a mouse embryo, there's the structure of the mouse egg cylinder, the trophoblast, extra embryonic endoderm, and epiblast that basically we're trying to recapitulate to sort of get this early embryo model in this case. And I aim to do this with epigenome editors. So these are Cas9 based tools that bind to the DNA, don't cut it, but instead you fuse them to transcriptional or epigenomic modifiers, so we can actually locally activate or repress elements of the DNA and do like functional DNA modifications. Part of the main reason for that is that we only use sort of the endogenous cell machinery to regulate endogenous vector elements to induce different cell types, and really have like a cell intrinsic based method to make cells of the early embryo, which has the advantage that we hope we didn't need to use any sort of media supplements to guide the cells into a specific phase. And of course, naturally, because it's a CRISPR-based system, we can also use this to do additional perturbations, like functional modifications, to follow up with functional experiments. So it's like a programmable device, use either perturbations or larger scale screens to study these embryo models in vitro. Basically, this is what I set up. So I have my embryonic stem cells on the bottom left here. I integrate transgenic DCAS9 VPR, so it's a transcriptional activator cassette that is a DOX-inducible system, so we can control the expression. And the second part is the lentiviral for as a guide RNA cassette, so we can actually direct the DCAS9 to our target. And again, this showing that without DOX, there's no activation. Plus DOX, we get activation, so we can choose when do we do this activation. Start at the end. We are flexible in that. And this is basically going back to like, well, we know that there are sort of three main cell types in the early embryo that we need to produce. Embryonic stem cells, we cannot get them for free because that's the cells that we start with. These sort form those epiblast or inner cell mass-like compartments. So the cells actually make the tissues in the adult embryo. And the cells that we have to make specifically from these embryonic stem cells are the trophoblast cells and the extra embryonic endoderm cells. And we know from previous studies that CDX2 and Gata6 are important for inducing those cell fates in the embryo. So we started out by inducing those factors and do we actually see changes that lead to those kind of cell types. This is some very simple validation. On the left, you have control guide RNA. On the right, we have CDX2 guide RNA. We see that we get CDX2 activation, no activation of control. And you'll see activation of a downstream gene called TFAP2C. So we do the CRISPR activation. We not only induce CDX2, but also downstream factors are getting activated, indicating there's sort of like a self activated program happening. And the same when we do our extra embryonic induction, extra embryonic endoderm induction with Gata6. Again, like no Gata6 in the control, but we see high levels of Gata6 with the Gata6 RNA and also the downstream FOXP1, FOXP2 activation. This is very promising. Our CRISPR activation seemed to work quite efficiently. But then, of course, the question is, do these cells organize into embryo-like structures? So have they formed sufficient unique features that they actually are able to form organized structures that mimic the embryo. And that's basically what we did here, is that we put these cells into agar-well plates. And these are multi-well plates that again have hundreds of microwells within each well. And the cells will sort of land in these microwells, and they start to form little balls of cells. So you can have 30 cells approximately in each of these microwells. Then the cells will start growing and interacting with each other. And we see the cells at time point 0. We add dogs. So at time point 0, all the cells are the same. We add dogs. The differentiation starts. 3 days later, collect the tissue, and we did immunofluorescent staining to see what happens. It is kind of what we get. So we label all of our nuclei, CDX2 for our trophoblast cells, OXO1-2 for the extraembryonic endoderm or primitive endoderm, and OX4, PUU5F1 for the epiblast, the merged combination. So we see that similar like the natural embryo, this FOXA1-2 is on the outside, CDX2 is on one end of the embryo, and OX4 is on the other side of the embryo. But a little bit more about the structure later in the talk. But we try to quantify some of these structures, actually get some measures like, well, how many cells do we get? So we can measure the number of cells per organoid by fully segmenting the full Z-stack of these 3D structures to get a sense of which kind of cells do we get and how many. But also some more complex features, like do we get formation of the embryonic cavity by measuring the signal of cells in the cross-section of these tissues. And for instance, using that, we find that 80% of our structures form this embryonic cavity, which is an essential step in embryogenesis. And sort of summarizing that is that we have this simple CRISPR-based method where we use epigenome editing to induce this X-cylinder structure without using any morphogens that we add to the media. So it's like completely cell intrinsic induction. We only need 2 elements, so we just need the GATA6 and the CDX2 elements to basically induce this self-constructing pattern. And there's some other examples here just to show for fun, like another cross-section here and sort of 3D representation on the bottom right here of what it looks like in comparison with the schematic of the natural embryo on top. That's one aspect, is comparing the structure. But another question is well, are these cells actually assembling on a molecular level the embryonic cells? So do these, for instance, show transcriptional similarities to the natural mouse embryo? And for that, we use single-cell RNA sequencing to find out, are the cells that we have in our CRISPR-programmed embryo models, or CPEMs, are they the same, similar to the natural mouse embryo? So with single-cell RNA sequencing, we find some distinct clusters, for instance, like extraembryonic endoderm, trophoblast cells, and epiblast cells in the model, which are sort of highlighted by canonical markers like GATA6 for extraembryonic endoderm, CDX2 for trophoblast, and then for the epiblast. And then I want to zoom in a little bit more on the comparison between the natural embryo. On the left here are a set of marker genes shown in our model, and on the right, same genes in the E5.25 mouse embryo. So actually mouse embryo data. And we find highly similar expression profiles of a set of marker genes for instance, the epiblast here in the purple highlighted box. For extraembryonic endoderm, where we do actually see also some subtypes of cells forming that, for instance, have one expression or LHX1 expression that is higher in cluster 1 compared to cluster 2. And we see that same kind of expression pattern in the natural embryo, showing that these groups are quite similar. And finally, the same for the trophoblast cells, where we see our CDX2 population upregulating several markers related to trophoblast cell development, such as TFAP2C and PDGFA, similar to the natural mouse embryo. So combined, this shows that this model both has transcriptional similarity and structural similarity to the mouse embryo. There was a great initial finding that we could pretty easily induce these cells, mix them together, and they self-organize into this X-cylinder-like structures. One thing we did observe from this data is that there seemed to be some level of epiblast markers left in our trophoblast compartment, in the CPEMs, compared to the natural mouse embryo. So I felt like, well, if we use this data to perhaps enhance some aspects of the model, like improve this trophoblast compartment, would it improve our efficiency of the overall model formation? And there could be like two reasons why we see this sort of little bit of epiblast expression there. Is this here? Okay. So do we see improve trophoblast compartment by either increasing differentiation time? So do we just need to induce CDX2 for longer to induce like proper or like more differentiated trophoblast cells? Or can we do the other way? We activate additional factors by multiplexing our CRISPR system to induce additional trophoblast cell factors that would enhance the differentiation of those cells. So we try to do both. So doing the pre-induction and the multiplexing approach. So our control is just like using our initial system with the CDX2 guide RNA, adding the offset sort of 10.0 when we add the cell to the agar well plate. And the second condition, we pre-induce this for 2 days with dogs before feeding the cells into the agar well. And then another condition is basically doing the same, but in this case, we also add a L5 guide RNA in the CDX2 cells. Now these cells are a dual guide RNA CDX2 plus L5 double activating system. And the reason why I choose L5 is because L5 is known to be a gatekeeper for trophoblast cell fate, and epigenetic repression of L5 in the embryonic cells, basically preventing this trophoblast cell differentiation. So I think this would be like a perfect candidate for us to study, because we can direct our CRISPR activation tool to the L5 locus to activate or like remove this epigenetic barrier, and then hypothesize that this would improve our differentiation of the trophoblast cells. Again, putting this in the same 3D model that we used before. Basically, this is what we get. So just looking at this pre-induction, I'm here just looking at the number of CPEMs, number of tissues that have a significant trophoblast cell compartment. And we found that if we just do the pre-induction with CDX2, so go from like a 25% to like a 40%, doubling our number of CPEMs that have like a proper TS number. Surprisingly, when we did the activation with CDX2 and L5 sort of at the time 0, we didn't see any changes. But doing this combination of the pre-induction plus the dual induction really like boosted that efficiency to 75%. So the combination of sort of the longer induction, moving that L5 barrier really seemed to significantly enhance the trophoblast cells we get. Highlighting that you can use this CRISPR multiplexing method to really tease out how these cells are formed in vitro and using them in this kind of models. And that's the question is like, well, just having the cell numbers is important, but are they actually localized into the right compartment? And to measure that, basically, I traced the signal in the X cylinder for both the OCT4 and the CDX2 staining, and plotted that sort of in this graph and clustered it based on the on that staining pattern in 3 different clusters. So in cluster 1 is basically kind of random. You have both like the CDX2 and OCT4 signals sort of intermixed, indicating there's no patterning. Cluster 2, there's like small domains of CDX2 positive cells, little green peak here. And cluster 3 is sort of like the real like proper separation of like a distinct CDX2 and a distinct OCT4 domain, kind of shown in the example here. And we find that when you do this pre-induction, the majority of the tissues actually get this sort of like highly organized structure in separate domains, where like the one end is CDX2 trophoblast cells, and the other end is the OCT4 epiblast cells. So this combination again is like really helping to properly organize our model in like the proper axis formation that we see in natural embryo. Again, to summarize this is that we're able to show distinct domains of CDX2 and opt for trophoblast and epiblast compartments. We could enhance this efficiency of this formation by doing the pre-induction and also like using multiplexing capacity to get the numbers and the organization matching the natural embryo formation. And the final aspect I want to highlight of this is, can we use this for any functional aspects? So I think that is maybe the powerful tool that we can hopefully do in the long term, is actually do more functional experiments, like observing certain pathways, certain genes, certain like risk factors in embryonic development, and see how this affect the development of our model, and linking that to actual real natural embryo development.
1 minute.
Thank you. So we chose to work on the basement membrane for this. The basement membrane is a structure that's in between the extraembryonic endoderm and the other cell types. So it's sort of this blue like line that is sort of like a layer of extracellular matrix that is sort of separating the different cell types. And this is our natural embryo, and again, we find this in our model. We quantify this where you sort of see this like green graph. It's like in between the Gata-6 on one end, the OX4 on one end, laminin is like nicely wedged in the middle. Sort of when you take this cross section of the embryo, we can measure them. And we also know that this laminin is highly produced by this extraembryonic endoderm cells. So these outside cells, they sort of pump a lot of laminin into their environment. And we also know that as epiblast cells, they sort of try to counteract that. At some point, they will like break down this matrix because they need to sort of make space to expand and cells to migrate through gastrulation. So there's like a continuous sort of dynamic, like establishing an ECM to maintain a structure, but also like degrading locally some of this ECM structure, so like cells can migrate and move to their proper location. It's quite a dynamic process and interesting to study. So we thought like, well, we can take advantage of our ES cells that currently have a control guide RNA only. And we can use CRISPR-A to either overexpress laminin by targeting the laminin alpha-1 gene, sort of let the cell pump out more laminin that they're supposed to, or do the opposite, where we let the cells overexpress proteases that degrade this basement membrane matrix. These are metallo matrix proteases or MMPs. And because we produce guide RNAs in our ES cells, they're only sort of being activated in this sort of purple compartment of the cell. Basically, only one half of our model is like making these additional factors, like creating a localized effect. And again, we do laminin staining to quantify this. And what we find is that when we do laminin activation, we get increased integrity of this basement membrane, so there's like less gaps in this sort of continuous matrix surrounding those cells. And it was not affected when we did the MMP overexpression. But instead, when we did the MMP overexpression, the basement membrane was thinner, so it seemed to have the laminin overexpression or the MMP activation. They seem to have kind of reciprocal effects where, you know, the one is sort of strengthening it, the one is sort of weakening it, but they use sort of slightly different outcomes. And probably one exciting thing about that is that we actually saw a phenotype in the structure of the embryo structures, where when we strengthened the basement membrane, our embryo models had a more round structure by measuring the difference between the long and the short axis of the overall shape, suggesting they have some kind of like block in their elongation in the axon there. Whereas if we weakened the basement membrane, this elongation seemed to be promoted. So the actual embryos seemed to get a little bit sort of like longer shaped prototype because they didn't have this barrier that was like preventing their growth as much. I guess the summary of this CRISPR perturbation experiment. Just to quickly sum up, in future work, we're interested in comparing this both in human and a mouse system to see what are some human-specific aspects of how these cells are developing and how this rates like implantation defects. You can use like CRISPR tools to really like dig into the specific pathways that are regulated in this mechanism. By also introducing, for instance, like orthogonal CRISPR systems, so you can combine a CRISPR activation with a different CRISPR repressor system, to really have the flexibility to independently target different pathways within specific cell types and see how that relates to their development and interacting with their environment. And with that, I want to thank all of my lab members and the contributors, especially Sayaka, who's an undergrad who did a lot of work on this paper and also a co-author on the paper. And of course, the funding sources from both NIH, firm, and my postdoc fellowship and WO Rubicon and the Genentech Kate Fellowship. With that, I'm happy to take any questions.
Thank you so much, Gerald. That was an excellent talk. Everyone, please put your questions in the Q&A box. And I'm going to go ahead and start us off. I was really excited about these basically mini embryos that you can make. And I was kind of curious how functional, you kind of touched on this a little bit with the MMPs and the basement membrane, but I was wondering how functional they were. For example, do they try and embed into the matrix that you're actually plating them on? Could you kind of use this as a proxy to study sort of the black box, I guess, if you will?
Yes, I think that is, so I'm not the only person who's working on this. Like other labs have tried some of these things where they try to like put these sort of embryo models in like a pseudo-pregnant mouse. And they find that they can implant, but then they don't really develop further. So there's something that is missing. We know there's some of the extra embryonic cell types that are missing, which probably lead to some like developmental block. So I think that is something that is currently unknown. Like, what do we need to do to sort of make them develop further, especially with high efficiency?
And that's really cool. Let's see, we have a question here from Michael Layden. Extremely cool. The transcriptional profiles are similar, but not identical. In particular, all cells seem to have higher expression of all the genes you showed. How do you interpret the differences between embryos and engineered systems? And do the differences allow you to predict what the limits are for how the engineered systems can be used to infer the biology of the embryos?
Yes, I think that's a great question because we have this natural data to compare to. So in our case, we did use the L5 as one example to really improve our model because the L5 was like lower compared. The L5 in our model was lower compared to the natural embryos, and well, this is really something that is potentially lacking in the engineered cells. And yes, I think the levels are important, and I think having more control over those levels could indeed lead to more robust formation of these different cells, and also the timing of it. I think it is known that some of these transcripts are turned on at a certain time, and they are repressed at a certain time. So having control over that would definitely be helpful to sort of more like specifically induce the cells, especially if you take like time course data into consideration. So you can take like the day 4, day 5, day 6, day 7 mouse embryo and really like figure out, okay, like this factor needs to be on for like 1 or 2 days, so then we need to shut it off. So you can really like engineer the circuits to sort of mimic that temporal and level aspects in vitro, but really leveraging that natural data that we have now for both, I think, mouse and also for human data. There is actually some data out there that we can use for them.
Thanks. And I actually had just another question about culturing the synthetic embryos. How far, I know you said that some people have actually tried to implant them in a pseudo-pregnant mouse. How far are you able to take them while they're actually in culture? What embryonic stage would you say you could get them to?
So it's like, I think we get most similar to this E5.25 mouse embryo. We do see like some excess formation in the sense that we see in about like 25% of our structures, we see, for instance, localized expression of LEFT1, TKK1, and SIR1, which are pattern effectors that that will start to induce like another axis formation in the mouse embryo and start gestulation around like E5.5 to E6. So we haven't gotten there quite yet. So all this stuff has been on sort of E5.25 timing. And I think what other labs have shown is that if you want to go further, there's definitely the sort of like bottleneck in efficiency, where it gets harder to sort of maintain this like proper axis formation into like the more complex structures of both the gestulation and like further advanced structures. And I think the latest structures that have been made in vitro from mouse are they've grown them, I think, for about 8 days, and they do see like some like organs forming like brain, like other organs in these structures. But I think the efficiency is something that is probably like the main thing. Is like, well, how do you make this differentiation robust? Where we start out with maybe like some factors we can control that at some point lead to maybe like aberrant development. So how can we control these factors like, during different parts of the differentiation, so we can keep sort of the differentiation in the right trajectory? I think that is the challenge in that.
The challenge and the excitement. Well, thank you so much. And if there are no other questions, I just want to thank one more time Drs. Hatkevich and Dr. Lodewijk for your excellent talks. I did want to let everyone know that this seminar is being recorded and will be available on the SDB website next week. Please join us next month for our seminar on Friday, May 9th, when Taylor Medwig-Kenny from the University of North Carolina at Chapel Hill and Ru Yen from Harvard Medical School will present. Thank you all for attending.